Hello, everyone, and welcome to today's Keynote webinar titled Precision Medicine as a Population Health Strategy. This webinar is a part of the 11th annual event in the Genetics Week virtual event series. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots. Before we begin, I would like to remind everyone that this event is interactive. We encourage you first to participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby within the event. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Please click on the Continuing Education Credits window located in the Abstract tab on the menu at the left of your screen and follow the process to obtain your credits. And with that, I'd like to now welcome today's speaker, Dr. Howard McLeod, Director, Center for Precision Medicine and Functional Genomics. For a complete biography on our speaker, please click on the presenter window that is located directly below the media player. Dr. Howard, you may now begin your presentation.
Thank you, Kaylee. Really appreciate the opportunity to speak with this group, and it's an exciting time to be in the field and see the progress that's going on, and really appreciate all the effort that LabRoots has done to increase that. Some disclosures, I am on the board of directors of some companies that are listed here. I'm a scientific advisor to several companies. I've been a speaker for Illumina, and I'm in debt to my wife. The clinical problem that we're tackling, at least on the therapeutic aspects of it, is partly because of the complexity that we've generated ourselves, and it's a good problem. You know, on the therapy side, we now have multiple active treatment regimens for most diseases. Now, there are still plenty of diseases where there's no treatment, and we need to have something there. But amongst common diseases, we often have tens, or in some cases, hundreds of options to choose from. And so something like high blood pressure, at least in the United States, there's over 100 different FDA-approved drugs or drug combinations that are available to treat a patient. And so, how does one choose that? How does one go forward if we're trying to manage health and do the best we can? Variation in response is still normal, and so whether we're treating disease or using therapy, we see some patients do well and some do not. Toxicity is also something that remains relatively unpredictable, although it's getting better in terms of being able to predict that. And then, of course, there's the issue that none of us want to talk about, and that is that the modern therapies, especially those that are highly specific and highly active, are very expensive. And so, how does one take advantage at a population level of these innovations when we really need to also be responsible over the use of the funds that are available? So, you know, difficult challenges there as we're going forward, thinking about the population. I want to remind us that probability is a main driver of our choices with health. We look at whether someone has a high risk of getting an infection, such as a viral infection, and decide, well, maybe we should vaccinate this child, vaccinate this adult. It's all around the probability of a bad thing happening. And there are some people that play the odds. They choose to maybe not vaccinate a child or vaccinate someone else. And in some cases, that comes back to be an issue because of the probability that is there. Within therapy, similar problems. We don't wait until someone has gone blind or has lost a kidney or a toe from diabetes. You see uncontrolled glucose, you try exercise, you try diet. If needed, you try medical therapy. Try to decrease the probability that uncontrolled glucose will result in some of these bad effects down the road. Same with statins for primary or secondary prevention for cardiac disease. Same with antihypertensives for stroke or kidney failure. The idea that we try to look at what is the probability of a bad thing happening and decrease that probability is a signature feature of the way we approach wellness as well as treatment of active disease. And the reason I put that picture down at the bottom is often what we need is the gutter guards or bumpers, I guess they're officially called. If you take a child bowling, often you can flip up those little guards. It doesn't mean the child will get a strike when they bowl the ball down the lane, but it means that the ball is not going to fall in the gutter, and it is more likely to at least hit some pins. And so often with precision health and with the probability work that's happening, we're trying to increase the probability of a good thing happening, trying to decrease the probability of a bad thing happening as we go forward. We'd use the same principle with disease. With in this case, colorectal cancer screening. You reach a certain age, your probability of the disease goes up. Therefore, you can intervene with colonoscopy, intervene with stool-based DNA detection. Now there's some new data coming out where some blood tests might be as good as these approaches, or at least near that. But the increased probability causes one to intervene, and this, by belaboring this point, what I'm trying to just remind us is we don't have to be perfect. There are very few examples, maybe if any, where we know exactly what to do 100% of the time. But if we see heightened probability, we can act, we can intervene, we can do something about it. And so that's going to be a feature as we're thinking about how to use genomics and other tools to improve the health of populations. Now, I'm going to focus in on drugs for a couple reasons. The main reason is because that's where I've spent a lot of my energy over the years, and so that's the area I know the best. But also, medications are a great thing and a bad thing. So adverse drug events this last year were the 5th leading cause of death in the United States. It looks like it's going to go back up to being the 4th leading cause of death now that COVID has diminished its mortality to enough. But the idea that medications are a major source of morbidity and mortality is something that is often ignored, not really recognized as being an issue, and one that we really just can't afford to ignore as we're going forward. Now, it's not one particular medication, or else that would be taken off the market. It's a collection of adverse events that cause these problems, but it is an issue that can be tackled. In countries like the United States, it's a heavily litigated issue, so there's that issue that we like to remove. As I mentioned before, more and more of these adverse events are now predictable, and so one can know about them ahead of time and intervene and go forward that way. And then these new therapies are quite expensive, and so how do we deal with all these things together? And really, the probability of success is one of the key features there. And if you take an example, this is a group of medicines that are approved for the treatment of cancer, approved in the United States for the treatment of cancers. All of them that are shown here are associated with a particular gene. And so if you have a gene variant that is associated with drug activity, that drug has a high probability of success. And so while there might be an approved indication, often they can be used across different indications because of their success. Now, if you're asking, the reason why these are different colors is because I have very poor PowerPoint pasting skills. And so at some point in time, I'll have one of my children fix it so they're the same colors. But the idea that we have a menu of options, any of which could be used if we know the right gene to follow, is the principle there. And the way we're taught is as if we're a Michelin star restaurant, and we if you've had the good fortune of being to one of these restaurants, there is no menu. You show up, you're giving a list of what you'll receive. It's usually 27 different items. They're all this big, and they taste amazing. And at the end, you get a massive bill for your experience. And that's similar to healthcare. You come in, you don't really know what's going to happen. You're given a bunch of items, you get a massive bill at the end. But the way we really are is much more like a buffet. When we look at how to treat a patient, especially with common disorders, there are a number of different options that all have good data behind them. We need to be able to pick one. And to date, we've picked them because of cost or access or oral versus IV administration, that sort of thing. But now we're seeing a lot more of the molecular data, understanding that this drug has a much higher probability of success because of the patient's genome and other features like that can be taken advantage. And so that's a major theme underlying a lot of what we're going to be talking about. The other piece I want to remind us is we also need to think about where we're getting our data. If you were to ask a fan of a particular sports team, what are the best sports teams in that particular sport, you're going to hear a lot about their favorite, and not so much about any of the other teams that are in that same league. The same is true with genomics. We've done a lot of studies, and in terms of drug development, we've done a lot of studies in one population, and not as many in others, and therefore we know a lot more about that. And this is some data that Dr. Todd Knepper and myself published almost 5 years ago now, looking across an 18-year period of FDA drug approvals. And what was identified on the right-hand side is depicting the different countries that were involved. And basically the punchline is, over this 18-year period, the number of countries doubled from 15, on average 15 countries contributing to the safety and efficacy data for a medicine, up to around 30 countries rather, contributing to that. Well, during that same time period, you see that the ethnicity and the race and ethnicity data really did not change very much. It went from 92% white down to 86% white. Well, in the United States, we're about half white in terms of the country, a little bit more than that. In the world, only 16% of the world population is white. And so if you look at this data, we know a lot about white patients in terms of safety and efficacy. We don't know much about many other patients in the world in terms of how they're going to behave. Humans are human, so thankfully there's a lot of commonality, but there's still variation that occurs based on the different populations. And so that sort of information has been important, just as a reminder that we're looking at very robust but only partial view of the world safety information when we use medications. There has been efforts like the Pharmacogenetics for Every Nation initiative that have gone and looked across many different populations, looking at the genetic variation that's found in these populations. And these are just 3 different drug examples: the average warfarin dose, the risk of GI toxicity from an anti-malarial drug, the risk of muscle toxicity from a statin therapy. And as you glance at these, you can see there's lots of variation that are happening. In some cases, like with warfarin, in general, Africa needs a higher dose, Asia needs a lower dose. But even within different geographic locations, lots of variation that is occurring with the average. And then, of course, within a population, there's a whole spectrum of need. But it's just a reminder that really, we need to understand this sort of variation as we make plans for populations, whether it's for a country or whether it's for a city or whether it's for a health plan or a group of college students, whatever it might be. Understanding that variability is key. Now, before I launch into some more of the data around precision health, I just want to ask you a question that we'll come back to at the end. And that is, so those of you that are familiar, this is the golfer Tiger Woods, one of the best golfers in the world. And he has self-identified the data from his parents, African-American, Asian, and Native American. And so I just showed you some variation that was occurring across the globe in terms of genomics. What do we pick in terms of a starting point for Mr. Woods? What's the best thing for him? And we'll come back to that point towards the very end. Now, in terms of precision health, one of the key features is that this is not genomics. Precision health is really embracing complexity. And what I mean by that is we can use genomics as a way of understanding the individual, but also their lifestyle, their environmental exposures, family history, other medical issues, all sorts of those things can come in and be important parts of the decision-making process. And for example, Nephi Walton, one of my colleagues in Utah, identified a large number of people who had a genetic risk for mature onset diabetes, of the mature onset diabetes of the young. They had the genetic risk for this, but many of them did not have diabetes. And the reason why is they had a very healthy lifestyle, to the point where they had functionally outrun diabetes, at least for the short term, because of the lifestyle component. And so it's, again, we like to keep things simple as humans. Reality is not. And so trying to embrace that is important as we're trying to do precision health. Now, I'm going to give you some data in the context of the Intermountain HealthCare System. This is a system that is based in the Mountain West of the United States. Its base is in Salt Lake City, in the blue dot in the middle of Upper Utah there. But there are around 400 clinics and approximately 33 hospitals in these 9 states that are that have some activity. And the motto of helping people live the healthiest lives possible, through precision health is the piece that one could add, for this. And so, as an example of where precision health is across the spectrum of life, at this particular health system, we see implementation of genomics in the neonatal intensive care unit, to try to very rapidly understand what is going on with a patient and how one could intervene. You see non-invasive prenatal testing, of course, which has become a normal way, at least in the United States, of managing these a pregnant woman's the pregnancy. Whole genome sequencing is being applied in a number of areas now, including adult cardiology, adult neurology, and as I'll get to, population health. And then, of course, there's targeted data that we'll talk on very briefly, whether that's targeting the cancer, or viruses or bacteria, or in the case of donor and recipient for a liver and kidney transplant. And then across the top is pharmacogenomics. And we're really seeing that across the whole spectrum of life. Everything from which medicine to use in a neonatal setting, all the way through to which pain control to use in a hospice setting. So literally the spectrum of life has some application of genomics today, and that is growing as the data emerged. And so genomics is really, part of the reason it's the centerpiece of a lot of the precision health efforts is because of this wide areas of application that have occurred. And many people are thinking, well, genomics isn't really applicable yet. Well, maybe not in your area, but it's certainly being applied in a lot of areas. And so there's a lot of interest in trying to get it right. Now, in terms of the pharmacogenomic piece, the drug piece, lots of different examples that are out there. I'm sure I'm missing some of your favorites on this list, but these are examples where the information is the dosing and administration section of the FDA drug label, the package insert, as it's often referred to. Now, there's about 170 drugs where genetic information is somewhere on that label, but this is the dosing and administration section. This is the one that really matters in terms of applying genomics. And you can see the top half of this list is mainly things found in the tumor, and that can be used to try to guide therapy for a particular cancer. And if you have one of these variants, it means that drug has a higher probability of working, and therefore can be applied in that manner. In the bottom list is a number of other areas, some cancer, some not, where there can be application that is occurring. And one of the things that has really changed over time is the use of this in the cancer setting. And at least in countries like the United States, most patients with metastatic disease, should be all patients with metastatic disease, but it's in most, and some with advanced disease otherwise, a piece of the cancer is obtained either through a biopsy, a fine needle aspirate, or a core biopsy, or through a liquid biopsy where one can take blood and look at tumor cells, tumor DNA there. Genomic analysis is performed, and then you can see what are the genes that are altered. And often, but not always, that will tell you some drivers of the cancer and some therapeutic opportunities. So in this list, HER2 or ERB2, as it's also known, is one of the genes that is altered. And there's a drug or a set of drugs that can be applied in this context. For the second gene, TP53 or P53 is altered, very commonly altered in cancers. But at the moment, there are no drugs that are available for that. There are some in clinical trial, but there's no drugs that are available now. And so this idea that one can look at what's driving the cancer and act on it accordingly is becoming the normal paradigm for this. And it's kind of seen in the print down below. A little bit more than half the time, genomic analysis can show what is broken, what is wrong in that tumor, and we can try to fix it. 100% of the time, it changes or refines the clinical decision making. And that's because if HER2 or ERB2 is not altered, these therapies are not options for this patient. And so knowing this data adds and subtracts options as we're trying to manage the patient's care. And so that's a very real and very practical way of this application. And, we all have these types of stories. This is a 56-year-old man with metastatic lung cancer, relatively young. He had some young children. He'd gone through all of the standard therapies, and they'd bought him some time, but did not really control his disease adequately. And there was no clear next option. So he was fit. He wanted to be treated. He needed to be treated in his mind because of the family responsibilities, and he wanted to move forward. And so genomic analysis was performed on a piece of his tumor, and there was an unusual gene variation. So the BRAF gene is not an unusual gene. It's altered in many cancers, but the particular variation in his genome was not a standard one you see often. And so there was no clear guidance from that. And so he was seen at the molecular tumor board where a discussion could happen. You look more deeply at the data. And it was identified that there is therapy, in this case, a MEK inhibitor, that was initially developed for the treatment of melanoma, would be active in his case. So here's a lung cancer who has run out of options, being treated with a melanoma therapy. And because of the specificity and because of the thoughtfulness around it, in this case, his tumor shrank significantly. There's still some evidence of disease, but it has bought him multiple years now, and hopefully will buy him multiple more as he's able to help his children grow up, help his the life of his family go in that way. And so these types of examples at the individual level are occurring, and therefore are driving a lot of the interest in these therapies. Stepping back and looking at a population, what other features are there that would make this worth doing at a population level? Well, this is some data from Derek Haslam and others at Intermountain HealthCare, looking at patients in a similar situation to this man. It's patients who have gone through all of the standard therapies, all of the on-pathway therapies, the guideline therapies, the FDA-labeled therapies. They still want to be treated. What helps? So this is a comparison of patients with solid tumors in that clinical scenario who got either the traditional clinician best guess or targeted therapy driven by molecular analysis. And what was seen as the targeted group had twice the survival as the other group. Still not long enough. We still want the survival to be longer than it is now, but demonstrating a clear benefit of this kind of therapy. And as it turns out, the economics were also favorable. So this is the cost per week per patient per week. And you see there's about $734 less to manage a patient in the precision medicine arm. And the reason why is the therapy is working better. And so they're not going to the urgent care, to the emergency department, being admitted to the hospital. So there is the extra cost of testing, but there's also the benefits that occur because of that. And so again, at the population and the medical decision-making level, one can make policy and can be thinking about populations using this kind of data while we're hoping for the scenario I just showed you about in terms of each of the individual patients. Now, looking at this list of pharmacogenes, you might have noted that there's a lot of things that are commonly used in all different areas of medicine. Pain control is one of the major reasons why someone seeks medical care. And it could be pain from a sprained ankle, it could be pain from a bone metastases, it could be pain from all sorts of things in between. And so that's one example where pharmacogenomics is involved. Vomiting, whether it's anesthesia risk or chemotherapy risk, antidepressants are commonly used across areas, and the list goes on. And so the idea that a spectrum of patients seen in a general medical setting, a primary care setting, for example, there is relevance now for pharmacogenomics across the area, not just in the specialty areas like oncology, where it used to be the key thing. And as we look at these examples, lots of individual examples. In this case, there's national or international guidelines that have been put forward. And so it's no longer something someone might do as experimental approach. There's now guidelines, well established, careful guidelines that have been developed. There's all these individual examples, and then one can start putting them together. And instead of looking at one at a time, really look at a panel that allows one to look broadly. And this, I don't know why Arnold Schwarzenegger was wearing blinkers for here, but and those of you that are not into horses, blinders is when the horse can't see or can barely see. Blinkers is when you focus their vision. And in this case, if you have blinkers on, you might only look at one gene, one drug, when really there's a whole opportunity to be managing the patient in a much more careful manner. And so when you look at some data from Sir Professor Pir Mohammed from Liverpool, published a few months ago, looking at populations, you can see a spectrum of different countries shown here. Well into the 90% of patients have a genetic variant relevant for pharmacogenomics. And so this is not something where you test the population and a rare individual might have an issue. This is something where you test a population and nearly everyone has something to talk about in terms of trying to better manage their care. And so it's a high yield exercise going forward when you look at a panel of variants. And there's now some really nice data that have come out. This is data from Jesse Sven out of Leiden and a number of European colleagues that are shown here, and lots of that aren't shown here. But the bottom line is that when you use a pharmacogenomics panel to manage general internal medicine patients, within a 12-week period, you can see a significant difference in terms of avoiding adverse drug reactions. And then along with that, the data will be coming out looking at issues such as economics and mortality and morbidity and that sort of thing from this. But the idea that one can use a panel in a group of patients and get a relatively quick return on investment within 4 months, or within 3 months rather, one can see the benefits, is really quite remarkable in medicine, and showing that panel testing for pharmacogenomics is an exciting population endeavor, as we're trying to really look and say, how do we mitigate risk as we're trying to manage patients. Now, the last little piece on pharmacogenomics before I shift into the final bit around disease genomics, population health, is that the financial impact is also developing some data. I showed you the relevance of finances for cancer screening. This is some data from our colleagues at the Kentucky Retired Teachers Association. Really a phenomenal group of leaders that run this health insurance for retired teachers in the state of Kentucky. Very proactive in terms of trying to look at things that would help their retired teachers have better health. And they did a study that was published fairly recently, where they looked at pharmacogenomics in about 5,000 of their retired teachers, compared to about 22,000 who did not get pharmacogenomics. And what was found was a reduction of about $2,500, a little bit more than that, in direct medical costs per year. And the benefit there was not just in terms of medicines, but again, medicines now working, so or not causing the toxicity. Either of those things kept people out of their urgent care, out of the emergency department, out of the hospital, reducing the costs for managing their care. And so this concept that intervening, making better medical decisions, and following on them is actually not only good for the patient, but also good for the population, whoever's managing that population, whether it be a government or a employer or some other group. We did a quick analysis looking at across Intermountain HealthCare at the patients who would have met eligibility criteria for the Kentucky study. There were about 180,000 patients that met that criteria. We then assumed that only 10% of them would take up the test. We realized that's low, but we tried to be conservative. We also predicted that the savings would not be as high as this, so we wanted to be conservative, and that suggested around a $47 million worth of savings that would be occurring in the first year by applying this sort of data. And so, again, if you're doing population health, precision health at a population level, one needs to think about the whole issues. One needs to think about how do we impact the individual, but also how do we impact the whole, not just the parts. And this idea that we can do something that is good for an individual and favorable for the entire system is an emerging theme that we're seeing in a lot of application of genomics. Now, shifting over for the next little bit, and then want to leave plenty of time for questions. One can also look at populations in terms of disease and what that means. And this is a study that Lincoln Nadal and a number of others put together across the Intermountain HealthCare System. It's called Heredigene, a really a beautiful study that went in and enrolled patients, and hopefully this is visible to you. Patients were enrolled in 3 different methods. They were enrolled a kind of a traditional way. You came into your doctor, came into the lab. You were then asked, do you want to participate? You were given informed consent. You signed consent. Then some of the blood that was obtained from you was then used to make DNA, to do whole genome analyses, both the chip and the sequencing versions of that, and then bring it back accordingly in terms of trying to apply it to someone's care. There's also a method I'll show on the far right of digital enrollment. So you might be watching a YouTube video of a cat and suddenly an ad pops up for the Heredigene study. You click on it, you can go online and enroll in the study. And then it tells you where you can go and drop off your blood sample, to give a blood sample and then enroll that way. The one in the middle was the most successful and is the most successful, and that is using residual blood. And so in this case, a patient comes in, sees the doctor, goes to urgent care, goes to the emergency department, is admitted to the hospital. Any of those scenarios, blood is obtained. Some of it is used for the required tests, and then there's some that's left over. For the patients with leftover blood, which is most, they've consented to be contacted as part of their conditions of care. And so they're contacted through the method that they preferred. It could be text, could be phone, could be mail, and they are then enrolled on the trial and do online electronic consent. And these different methods have allowed about 180,000 patients to be enrolled over the last few years. They're going to be stopping at about 250,000 and looking and seeing whether there's relevance to go forward. But this idea that one can look at a broad population of patients is really an important step forward. Now, amongst the patients that have been enrolled, you can see the average age. It goes from newborns all the way up to 90 plus years old. There's patients from most United States. The state of Delaware is, I believe, now has been obtained, but that was the missing one. It's been in a general population setting and a specialty setting, so it's not just primary care and it's not just oncology or cardiology, et cetera. And there's also inclusion of some of those that are often excluded, and I'll show you that in just a moment in terms of some of the groups, Native American, for example, that are also included. But also half of this study are patients from rural areas. And those patients are often just neglected in terms of their contribution to to data. You can see a map of, even though the enrollment was all done along in Utah, there's patients from all around. Maybe people who are coming to visit Zion National Park, or people who are visiting their relatives or whatever, but people have enrolled from all sorts of different countries. Now, an important part as well, as we'll get to in a second, is it's not just research. Some of the clear gene risk data is being obtained, confirmed, and there are thousands of patients who are at risk now, who are being identified, and intervention is happening, as I'll show you in just one moment. So this is just a snapshot of the first 100,000 participants or so. You can see it's a very white population, about 92% white, that is reflecting the region that of this area. But there is enrollment of individuals, Native American and Pacific Islander in particular. Now, while the percentage is low, the inclusion of almost 600 of each of these groups allows us to now start getting a clearer look at the risk that is present, realizing that there's almost no data out there for many of these things that we're looking at. So it's not that this is a great number, it's just that it's better than nothing. My favorite saying is, in the land of the blind, the one-eyed man is king. So we have at least some view of what's happening, whereas before we had nothing. But you can see the spectrum there in terms of different racial and ethnic priorities. Genomically, and this is just looking at 1,000 random patients from that 100,000. You can see that the self-declared race self-reported race is listed there. And then there's a fair amount of genetic variation within those groups. So some of them more than others. But the idea that one can look at these different features is certainly a part of it. Now, a critical piece that Nephi Walton and Bryce Christensen and others put together is taking the research findings and turning that into a clinical intervention. And so there's a lot of informatics work that was done, a lot of individuals helping evaluate these areas. A key piece of it is looking at the risk lists. And this gene list is small. It's not meant to be necessarily something you can read easily, although you can look at the copy of the slides and see it. But the important thing is there's a number of genes where there are known variants that are pathogenic, definitely cause this disease to happen. And so some of these are cancer risk, some of these are cardiac, some of these are metabolic, et cetera, as I'll show you in the next slide. But this priority list is the one that's being worked on as going forward in this area. And so again, this is from the first 25,000. Sorry for jumping around with the various numbers, but it was what was handy. You can see that lots of cardiac variants, lots of oncology variation. There's a big wedge just for hereditary hemochromatosis, which is an iron overload syndrome that, in its severest case, can cause liver transplant, need for liver transplant. And then there's a number of other different areas there. And so again, lots of variation that's occurring. Somewhere around 6% of the patients needed something significant, something where we would contact them to try to intervene. So it's not just trivial findings, but really medically important findings. The way things are managed is shown here in terms of hemochromatosis as an example. So you can check iron overload studies and ferritin in particular, and if the patient has a normal ferritin and no evidence of liver function test abnormalities, they can be rechecked annually by their general practitioner, their primary care physician. If there's elevation in ferritin, but liver function tests are normal, either the primary care physician or a gastroenterologist, a referral happens, and phlebotomy. This is medieval therapy, bloodletting, but phlebotomy occurs until the ferritin has come down into the normal range. In some important cases, patients have had an elevated ferritin and some evidence of liver function test abnormalities. This is a referral to hepatology, the specialist gastroenterologist, and intervention is happening there, again with phlebotomy, often twice a month initially, to try to bring that down. And we have a number of cases now where they presented with very high ferritin, some evidence of liver function test abnormalities, and after a couple of months of intensive phlebotomy, able to bring ferritin down to normal, LFTs start normalizing. As at least the opinion of our liver transplant surgeons, many of these patients were headed towards needing evaluation for liver transplant, instead are reverting back to something that's much easier to manage, much cheaper to manage, and can be managed in an outpatient type of manner. And so, again, these sorts of interventions at the population level allow the individuals to be better cared for while we're reducing the disease burden for the population as a whole. Last little piece on this part, and that is around the economics. And what you can see is, among the first 10,000 participants, here are the number of people that had these various disorders. Here's the actual charges for these patients that were associated with these. And these are the outpatient charges. We excluded inpatient charges or anything like that. But you can see that it's a significant amount of money that is being affected. On average, it's well into the 20s of thousands per patient that are associated with these events. And so, whether you're a fee-for-service health system where this was revenue, or whether you're a managed care type system where this can be cost avoidance in terms of further excess costs, you can start seeing the economics around this are quite significant in terms of doing a population-based approach. Now, one element is, if you look at a healthcare system such as Intermountain, I mentioned about the 33 hospitals and the 400 clinics. How does a small clinic in the middle of Montana or in the middle of Wyoming benefit from something that can happen at the big medical centers, at the big academic medical centers? And part of it is really trying to think about who delivers this care. So in terms of pharmacogenomics, over 90% of people have to have something done when they get tested. So this isn't the 6% I mentioned for disease. It's the 90+%. And so it can't be ignored. And there's very few specialists that are out there. We have things like polygenic risk scores and others that are coming along that are going to be applied. And these polygenic risk scores, they might be developed for breast cancer or for Crohn's disease or for your favorite disease. But the way they're going to be implemented is you're going to do one test and you're going to have 20 or 200 or a large number of polygenic risk scores related to diseases that will come back. And so who manages those? It's a cardiologist doesn't want to manage Crohn's disease. And so the idea that one can have these various problems to be dealt with is important. We have acquired areas like clonal hematopoiesis of indeterminate potential. Most physicians and advanced practice professionals really would prefer to do a consult or a referral type strategy. They don't want to have to keep up with yet another area that they weren't prepared for. And so we're seeing a number of really interesting models. One is we're seeing pharmacy-led clinics that are starting, where these patients are managed by pharmacy expertise. Even areas like polygenic risk score are being managed by these trained clinical pharmacists because they're more comfortable with applied genomics than most physicians are, or nurse practitioners or PAs. There are so few medical geneticists and genetic counselors that that's not really a viable option for most centers. When you have them, fantastic, that's plan A, but plan B is going to be the majority, and that is going to be a different type of specialist developing for the community. Telemedicine is also an important area. And so, for example, at Intermountain, 2 different types of clinics were developed. One was for high-risk cancers. And so, for example, if you had a BRCA2 variant, you have to see a breast specialist and a gastroenterologist and a gynecologist and a urologist if you're a male. That's a big hassle. Lots of different, how do you coordinate all that? Real problem. And so developing one clinic that is informed by all these specialties and then can be seen in one place. In this case, a telemedicine clinic is really has been really a boon, because now that person in the middle of Wyoming can get this high level care that they would not have been able to get otherwise because of geographic distance. Same with other areas of genomics. But the point is not these are the best clinics. The point is we need to be thinking creatively about how do we apply population health strategies, preferably with virtual care, because now you can democratize the availability of and access to care in ways that you can't do through the traditional brick and mortar type of approach. So just finishing things up here, and have plenty of time for questions. I mentioned what genomic profile applies to Tiger. If you're in the United States, you might think of him as not only one of the best golfers in the world, or used to be one of the best golfers in the world, but an African-American golfer. If you're in the rest of the world, you most likely think of him as an Asian golfer. And that's because he's 50% Thai, 25% African-American, 25% Native American. His mother is Thai. And so is he Asian? And therefore we do some of the Asian learning that I showed you from the genomics early on, or African or whatever. Which one of these applies best to him? Well, the real answer is test him. And so it's just a reminder that if we're doing population health strategies, ultimately they mean deriving data from the individual and then applying policy across the population, influenced by the individual patient's data. So population health strategies are not remove the individuality of a patient, but rather the opposite. Understand their needs and how it fits into the overarching population framework, and then be able to apply it accordingly. And so again, lots of opportunity going forward with that sort of thing. And so I'm going to stop at this point. Happy to answer questions. But the key points is that the application of genomics and environment and other features to understand health at the individual level or the population level is now. It's happening now. It's not something that maybe there's someday. It's going to be better tomorrow than it was today, but it is now. And so that's the opportunity that we're all dealing with. So I'll stop at this point. I want to thank you very much for your time and look forward to the questions that are coming up.
Wonderful. Well, thank you so much, Dr. Howard McLeod, for your informative presentation. We will now start the live Q&A portion of the webinar. So if you have a question that you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. All right, so we've already got some great questions coming in through the chat here. So we'll go ahead and dive right in. This first question asks, is access to genomics technology limiting for widespread application?
That's a great question. And there has been a time when if you didn't have the expertise in-house, really next door to your patient, then it might as well have been on the moon. I mean, it just was not a practical thing. Now with the logistics that has been built up, one can be at that clinic in Wyoming that I keep referring to, and yet access the most sophisticated technology by sending the sample over to Boston or wherever it needs to be, to St. George, Utah, to be analyzed and then go forward. And that idea of being able to really move on the technology, it doesn't have to be local. It can be wherever it needs to be, but then the application can be local. And so the technology piece, I think, is really advanced in terms of making it so that anyone can really access whole genome sequencing if they want to, and anything in between.
Great, thank you. We also got a comment in our Q&A box here, so we'd love to hear your response. This says the integrated use of PGS with other omics data is predicting clinical risk and the analysis of their interactions with environmental factors. This appears to be the road ahead for personalized medicine.
Yeah, so very good point. Looking at polygenic risk scores and features like that, bring in the genomic information, as I mentioned. So it might have as little as 5, as many as 1,000 genetic variants from the patient that contributes to the prediction. Typically adding environmental exposure or some other feature to the genomics heightens the predictability of these. And so we're definitely seeing some very nice data The NHGRI, the Genome Institute at the NIH in the United States, has funded a big program with Emerge and others to try to tackle this. And so the data is maturing, as the question mentioned, the comment mentioned, to really be something that's exciting and ready to go. The difficulty, and I alluded to this on one of my last slides, the issue is not that we will have these. The issue is who is prepared to apply them. And so what we're seeing is that it's not, when you send away a polygenic risk score, they're not going to be one at a time. You do one genomic analysis, you have the information to do all the polygenic risk scores that company can offer. And so it's really going to become a general medicine problem, not a highly specific disease risk problem. And so that part of it needs to be built up. And that's why I think there's these other groups that are emerging that are really going to be managing a lot of this, because there's a lot more of them available, and they're already looking at things like pharmacogenomics. And so adding additional genomics is going to be an easy step rather than retraining an entire workforce, which just won't happen.
Great. Thank you so much. Another question here for you. What will be the upcoming research topics in precision medicine?
Well, I think we've heard about the polygenic risk score activities, and that's certainly a big one. There's also a lot of effort around what other areas what other areas of risk can be brought in. And so while I've alluded to the inclusion of environment and such in polygenic risk scores, the specific analyses of environment are pretty rare. We're typically like, oh, someone is a smoker, or maybe you have pack years, and that brings it in a little bit more. But we really don't know a lot of detail. So I think we're seeing a lot of other omics that are going to be applied. Rather than using someone's age, why not use someone's methylation pattern, and have that, because that might be a better predictor of the acquisition of events over time than chronologic age. And so again, I think a lot of the emerging stuff is not just how do we use what we have, but how do we refine it so that it's better, it's more appropriate. And we're seeing a lot of algorithms where race is falling out of the algorithm, not because of political correctness, not because of fear of being canceled, but because we have a better marker that captures the true source of variability, so that race is really not relevant because there's this more precise marker. And so I think that's a lot of the research now, is how do we better refine these predictions? How do we understand what's behind the biology, and what is the biology rather behind the label, and then apply that accordingly.
Great, thank you. We're getting some great questions in here from our audience. This next question asked, have your studies included gender data within and across populations?
Yes, they do. Typically, those with genetics data, you can get sex and gender both as variables that are applied. There are some areas of disease risk, as well as disease management, where gender is an important variable in terms of how one manages that. And some of them are obvious things, like endocrine-based therapies for breast cancer risk, for example. There's a reason why patients with BRCA2 or BRCA1, only 1 in 1,000 breast cancers are male, and that's because some of the endocrine features that a female has heighten the risk, heighten the way that ends up presenting itself. But there's a lot of areas where we're finding there were supposed to be gender-based risks, but they're not. They're not found, and that's been important. A lot of areas of cardiac disease, for example, the gender differences are really not holding up. It's more the way that studies were done in some of these areas. And so I think that's an important element, is understanding objectively what is the contribution of gender, and what does one, how does one use that? Ultimately, it's the best care. If you're letting political correctness or some of these other things get in the way of how you're managing patient care, you need to really examine whether you're doing the patient a favor. And that's, I think, there's been things we've done for political reasons that have harmed patients and vice versa. And so I think one of the challenges for us in biomedical sciences is to let our opinions stop at the door and think, all right, what is the data? How do we help this patient? And then we can worry about the other stuff later.
Absolutely. Well, we are running a little bit low on time here, so we can ask one more question before we wrap up. I know AI has been a hot topic recently, so what are your predictions of the effect of AI on genomic profiling in precision medicine?
Yeah, so AI has some interesting potential. It is certainly a lot of the talk about it in healthcare is future tense. There are some areas like rapid scanning of large number of images, whether they're radiology images or pictures of skin for disease, cancer risk, whatever, where there's already benefit happening. Some of the tenets of patient management still have not appeared yet in AI, and so AI might be able to pass the exam to be a physician, but can't necessarily deliver care. But they can do a lot of heavy lifting. It could be simple things like access to information off hours, where your doctor may want to get some sleep, and so your nurse might want to get some sleep, and therefore the chatbot can be helpful in that case. All the way through to some of the AI mapping that's occurring to try to make sure better surgical resection happens or such. But right now, in terms of precision medicine, a lot of the AI has taken the data from maybe 20,000 feet down to 1,000 feet, but it needs to be down to ground level before it'll be widely applied. And so a lot of AI today in precision medicine is really more hype and more important for trying to get venture capital money. In reality, we're still a ways off in terms of a lot of patients being helped by AI on a daily basis, but we'll get there. It'll be important.
Absolutely. Well, Dr. Howard, thank you again so much for being here with us today. Do you have any final comments for our audience?
I think that just as you're thinking about the areas that you work, whether it's in research or in clinical or in marketing or in whatever it might be, think about how your world can be more precise and how we can try to expedite. We don't want to prematurely expedite technology, but this 17-year lag of innovation that has been observed over the years, a lot of that is a case of not really focusing on what is really moving the needle. And so I would encourage you to think about your area and how these innovations can happen faster so that people can benefit now rather than someday.
Great. Well, I want to give you one final thank you, Dr. Howard, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until May 18th, 2025. LabRoots will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.